This episode is all about virology, and it assumes you already have knowledge about genetics and cell biology. Be sure to listen to previous episodes, such as Fall Asleep to Genetics or Fall Asleep to Biochemistry, to refresh your knowledge on these topics if needed. Enjoy. Viruses are simple and only reproduce when they are associated with a host. Every virus must have a host cell, and it is highly likely that every cell is a potential virus host. The interaction between the virus and the host is highly specific. Cell surfaces feature proteins or protein complexes that perform other functions for the cell. Though these are not intended to be receptors, viruses use these proteins or protein complexes on the host cell's surface as a receptor. Through this receptor, the virus introduces an instruction set to the host. The host will then unintentionally adopt these instructions and begin to build more of the virus. This process of virus reproduction must be as efficient and rapid as possible. To be the most efficient, viruses are self-assembling and feature structural components that are polymeric. There are 4 components of a virus. These components are the protein capsid, the genome, the envelope, which is an optional component, and viral encoded proteins for structure and reproduction of the virus. The capsid is a container of sorts that is made out of proteins. Its job is to protect the viral genome, since the instructions to build new viruses are stored there, while also allowing the genome to be delivered to the host. Structures of capsids can range considerably. They come in a variety of shapes and sizes, and there is no correlation between the size and host cell type. The most common structures appear to be geometric in shape and are built from multiple capsid proteins. To the dismay of researchers, the size of the virus contributes to its effectiveness at avoiding filtration away from cells. It also means that viruses can only be observed using electron microscopes. The viral genome is quite varied in the type of nucleic acid and structure, more so than the genome of the host cells. Classification of viruses is based on the genome type, such as naked capsid or capsid surrounded by phospholipid membrane envelope, for example. The viral genome also contains cellular enzymes that encode for capsid proteins and any specific enzymology that the virus needs but the host does not normally make. The envelope is built using the host's machinery for protein synthesis. The virus cannot make the envelope on its own. Luckily for the virus, the envelope is not a necessary component, only an added benefit if able to be constructed. The viral encoded proteins that a virus contains are proteins or nucleic acids the virus involves when hijacking the host cell. When a virus is out in the wild, it is in its infection stage and is referred to as a virion. Depending on the machinery of the host cell when it was made, the virion may or may not have an envelope. Regardless of whether it is naked or enveloped, the capsid and nucleic acids will likely be the same. The difference is whether or not a lipid bilayer is present. Only enveloped virions will have a lipid bilayer. Atop the lipid bilayer of the viral envelope are proteins that play a role in host recognition. These proteins are encoded by the viral genome. The first virus ever detected was the tobacco mosaic virus, abbreviated TMV. This virus causes mottling of tobacco leaves, which was previously thought to be caused by a bacterial infectious agent. Early researchers tried to filter out what they thought was the offending bacteria by streaming the infected sap through porcelain filters, but their attempts were in vain. The sap remained infectious even after filtering. Using an electron microscope to study the sap, it turned out that the leaves were being affected by a non-bacterial agent, and instead the virus TMV. This virus has a simple, repeated capsid protein that is arranged as a helix, producing a long tube to contain the viral genome. The helical capsid is the simplest arrangement of virally encoded proteins to form a virion. Another common shape is the icosahedron, which is a polyhedron with 20 faces. The icosahedron is used by many viruses since it is very strong and easy to assemble. Each capsid unit is called a protomer. In icosahedron-shaped capsid, the protomer can be assembled into groups of 5, called pentons, or 6, called hexons. These subassemblies of protomers are referred to as capsomers. They arrange to produce the icosahedron's 20-sided shape. A combination of both the genome and the protein coat are called nucleocapsids. More complex viruses exist, such as bacterial viruses known as bacteriophages, eaters of bacteria. These viruses have a capsid and a helical tube or sheath, giving it the appearance of a hypodermic needle or lunar lander. The proteins of the tail fibers are virally encoded and allow the phage to come into contact with the bacterial envelope. The phage then injects its genome into the bacterium when the sheath contracts. All viruses have stages of viral infection, though they may not all be performed in the same way. Viruses must first recognize its specific host, then attach. They must then get their genome into the host. Viruses can do this by either staying on the exterior of the host and injecting the genome, such as bacteriophages, or the virion can enter the host while the genome is uncoded to become available to the host type or genome type. Viral proteins can then be synthesized by the host's ribosomes, but this happens in stages: early, middle, or late stage proteins. The virions then self-assemble and are released. If the host cell is destroyed, the release occurs immediately. Some virions may be released without killing its host cell, while others cause a slow death to the host cell over longer periods of time. In summary, the 5-step process of viral infection is: 1. The attachment of virus to the host cell. 2. Entry of the viral nucleocapsid or nucleic acids into the host cell. 3. Synthesis of viral proteins and nucleic acids using the host's pre-existing machinery for protein synthesis. 4. Self-assembly of virions. 5. Release of progeny virions. To make vaccines or therapeutic agents for viruses, the virus must be grown in the lab in order for its viral life cycle to be studied. This can be extremely difficult to do if the host cell is difficult to grow in culture. It is common for fertilized chicken eggs to be used as a surrogate system, as they allow the reproduction of many virus types. Chicken eggs also have a variety of structural features that are available for the viral life cycle to proceed. To study the virus, researchers will inject the virus into specific cavities in the egg that can best mimic those of the normal hosts. Some positions are better than others to allow the best opportunity for the virus to reproduce. Some of these positions include the chorioallantoic membrane used for the herpes simplex virus, pox virus, and Rous sarcoma virus, amniotic inoculation used to study the influenza and mumps viruses, the yolk sac used to study the herpes simplex virus, and the allantoic, which aided in the study of influenza, mumps, the Newcastle disease virus, and the avian adenovirus. The use of eggs has made it possible to develop many of the vaccines that are commonly available against human disease. Conducting viral assays can be difficult. Some viruses make holes known as plaques in the lawns of host cells. For example, to study the effect of E. coli injected with diluted bacteriophage solutions, the cells were added to a solution of soft agar, then poured onto a regular agar plate. As the cells grew on the lawn, they became infected with the virus, and a hole formed at the injection center. The cell is said to have been lysed. The lysing releases more viruses into the surrounding lawn, compounding the rate of cells lysing. Eventually, the lawn became confluent, and the cells stopped growing. When growth rate stops, so does the infection. The holes created in the cells can be counted like a colony to enumerate the virus. Another example of this is the poliovirus. It behaves the same way on lawns of monkey kidney cells. Cultured fibroblasts form confluent lawns of spindle-shaped cells. When they are infected with a virus, the cells in the lawn change their appearance. In this case, the death of the cell is obvious. Due to their small size, viruses are extremely difficult to purify. There is always some cell debris that is smaller in size than the virus particles, quite problematic for the purification process. The purification method is done via centrifugation, either differential or velocity-based. Differential centrifugation is when a mixture of virally infected cells, known as lysate, is placed in a centrifuge tube and spun at increasingly fast speeds. Cell components that are larger than the virus are removed at each step, resulting in an enrichment of the virus in solution. Velocity centrifugation seeks to purify the virus from similarly sized particles. The mixture is spun at high speeds through a gradient of sucrose solution to separate fractions with different mobilities. Slow sediment components rest on top, while faster components settle at the bottom. The virus is then collected by piercing the bottom of the tube and removing small amounts of sample without disturbing the gradient. Viruses are similar in density and sedimentation rates to the components of cells. The density of viruses is comparable to those of ribosomes, proteins, nuclei, chloroplasts, and starch granules. Viral sedimentation coefficients are similar to the coefficients of the endoplasmic reticulum, ribosome, DNA, RNA, and approximately comparable to some proteins. To measure the size of viruses, an electron microscope must be used. Standards such as 264 nanometer beads are employed because these are much larger than TMV, for example. Enumerating is also a difficult task, but it is made possible thanks to the electron microscope again, or plaque assays already discussed. Alternatively, if a disease results from the viral infection, the infectious dose can be determined, though this method is an indirect measure of the amount of virus present. In summary, virus assays are completed either by counting the particles, performing plaque assays, or quantifying the infectious dose. The taxonomy of viruses is another extremely difficult task because the grouping is highly unclear. Too many different characteristics could be used for sorting. For example, viruses could be grouped according to the nature of the host animal, plant, bacteria, insect, or fungi, by nucleic acid type, such as single-strand RNA or double-strand DNA, by molecular weight, by segmentation and the number of pieces of RNA, by its capsid symmetry, by the diameter of the capsid or nucleocapsid, by the presence or absence of an envelope, by its sensitivity to ether, by the number of capsomeres in icosahedral viruses, by the immunological properties, by the gene number and genomic map, by the intracellular location of virus replication, by the presence or absence of a DNA intermediate, such as single-strand RNA viruses, by the type of virus released, by the disease caused by the virus, by the special clinical features it has, or by the mode of transmission. There are simply too many ways viruses could be classified, so it is best to agree on a taxonomy method. The most commonly used classification method is the Baltimore virus classification. Different classes are grouped based on genome type to make it obvious whether genomes are made of DNA or RNA. Keep in mind that some genomes can contain double-stranded RNA that are decidedly different to the genomes of cells that are using double-stranded DNA. Retroviruses are the exception to the rules for DNA genomes. Retroviruses have RNA genomes, but they code for an enzyme which reverse transcribes RNA into DNA after it enters the host, hence the name retrovirus. Back to the Baltimore virus classification. In Group 1, we have double-stranded DNA containing viruses. These viruses transcribe DNA to RNA. Many viruses in this group make the host responsible for making the viral proteins. Many viruses in the group also encode their own version of the DNA polymerase that copies the viral DNA. In Group 2, we have single-stranded DNA viruses. These viruses make the complementary DNA to generate a double-stranded intermediate form that is used to make the messenger RNA needed for protein synthesis and more copies of the positive DNA to be used for virus assembly. Bacterial DNA polymerase converts the single-stranded DNA into its replicative form in a rolling circle. That replicative form can then be transcribed to mRNA, translated to proteins, and copied into new variants. Or, through rolling circle replication, it can be converted to single-stranded DNA and given to new variants. Group 3 contains the double-stranded RNA viruses. These viruses already have the template they need to make more of the mRNA-like strand, so they make more of the complementary strand to make more copies of the double-stranded genome. Group 4 has the positive single-stranded RNA viruses that are used directly to make proteins. These viruses behave like mRNA immediately after entering the host, making them very efficient viruses, as they can begin making proteins quicker than the other classes. Group 5, by contrast, has the negative single-stranded RNA that are not like messenger RNA. Thus, the RNA in these viruses is not used directly and must instead be copied into its complementary strand by an RNA-dependent RNA polymerase before it can be used. Group 6 covers the single-stranded RNA retroviruses. In these viruses, the single-stranded RNA is converted to double-stranded DNA by the virus's encoded reverse transcriptase. The reverse transcriptase breaks every rule human cells follow, so note that we do not have an enzyme like this in our bodies. After the viral genome is made into double-stranded DNA, it can be transcribed into the single-stranded messenger RNA that makes viral proteins and become the packaged viral genome. When in the double-stranded DNA form, the virus can integrate into the genome of the host. If this happens, it can persist there and be passively replicated as long as the host remains capable of reproducing. Messenger RNA activity converts the positive strand of RNA into RNA replicase. The positive strand can be converted into its replicative form and back again through RNA replicase. Translation moves the information from the positive RNA strand to viral proteins and new variants. Finally, the double-stranded DNA retroviruses belong in Group 7. Double-stranded DNA proceeds through an RNA intermediate before it is reverse transcribed back into DNA. Single-stranded RNA is packaged into the capsid, and the RNA is reverse transcribed back into DNA before the virus is released. An example of a virus belonging to this group is the hepatitis B virus. Something to note before we move on from the classification system is that for both the positive and negative strand RNA genomes, it is impossible to make more of the RNA genome without an RNA template. A template of RNA is always needed to make copies of the complementary strand, and this is a must to make more viruses. Also note that in eukaryotic cells, double-stranded RNA viruses do not have to enter the nucleus, but because all DNA replication takes place in the nucleus, the double-stranded DNA virus must get its genome into the nucleus in order for it to be reproduced and subsequently exported to the cytoplasm for virus assembly. I mentioned bacteriophages before, so let us return to it briefly. Recall that bacteriophages are bacterial viruses. Most of the different Baltimore classifications are represented in bacteriophages, and icosahedral capsids are common, though other shapes are also possible. An example of a bacteriophage is the phage T4 that was isolated from raw sewage in 1930. The phage was attached to E. coli and quickly became the most studied virus of all time. Its genome is quite large, containing about 160 genes. The double-stranded DNA viruses of E. coli were the first viruses to be studied in detail and revealed the first recordings of viral life cycles and infection timeframes. Max Delbruck is the notable researcher in this case. He conducted a one-step growth experiment that revealed a single round of the T4 phage infection. Just a single step of growth allowed for observation of phage assembly to be made, and the time required to complete each step to be recorded. Keep in mind that this experiment was done in 1939, well before the invention of the electron microscope, so plaque assays were used to detect the assembly of the active phage. The procedure followed was to infect the E. coli with the phage and then rapidly dilute the culture to prevent successive rounds of infection. With this method, only the first round would be studied. Studies of the virus revealed a latent period during which no phage was seen outside the cell, because no active phages were found in the supernatant following centrifugation. Thus, the infected cells could be pelleted after being separated. Results showed that the phage is released about 20 minutes after infection, with 60 phages per cell for this virus. Following the latent phase is the rise period, when the virus is detected in the outside cells. This is the period of time where the assembly of the phage takes place inside the cell. The phage can be liberated and counted by breaking the cells open. The plaque assays are performed on the liberated phage. The eclipse period is the period of time after infection when no virus is assembled. Although out of order from my explanation, the eclipse period actually takes place at the beginning of the experiment, even though the synthesis of components is underway. To reiterate, it took 20 minutes to make 60 copies of the single infecting virus from the injected genome with this method. After the electron microscope was invented, it became possible to perform the one-step growth experiment and observe the phage inside the cells directly. Returning now to the life cycle of viruses. An important stage in the cycle is when the virus attaches to cells. This is called adsorption. Adsorption always requires the virus to interact with a cell surface receptor. Note that different viruses recognize different receptors. Usually, the receptors are proteins in the membrane, but they can also sometimes be proteins found in the flagella or pili. The mechanism that drives genome delivery is very complex and is not yet completely understood. An observable step in the process is the contraction of the tail, but this may not necessarily be the force that actually drives the phage genome into the host. There can be a multiplicity of infection in which one cell can have many phages on its surface. Normally, though, phage have exclusion mechanisms so that only one genome is allowed to infect the cells. So, despite the interaction with many phage, only one can be successful in injecting its genome. A bacteriophage called lambda has proven to be extremely useful in viral studies. Historically a research organism, lambda has a smaller genome and a spherical trick that allows it to do what T4 cannot. It is a temperate phage, meaning it is capable of lysogeny, to have a non-lytic relationship with its host. After it infects E. coli, lambda decides whether it can go through a lytic cycle or become integrated into the genome, which is what lysogeny means. Lysogeny takes place when the infected cell is not growing optimally for the lytic infection. When chosen, lambda can integrate a copy of itself into the host genome at a particular position, known as the lambda attachment site. The virus then enters the prophage state, and an integrated copy will be replicated passively as the cell grows. When the cells are growing optimally for phage reproduction, lysogeny can be replaced by the lytic phase. To reiterate, lysogeny is when the viral genome, the prophage, is integrated into the host DNA and is replicated without killing or lysing the host cell. These cells are said to be lysogenic and can also be called lysogens. Note that the host cell cannot be superinfected by a virus of the same type. In other words, it has immunity to superinfection. Lambda also has a cluster of transcriptional promoters. These promoters can either face to the left or right depending on the prominent transcription direction that is regulated in part by the host cell's proteins. Rightward transcription leads to production of gene products for the lytic phase, while leftward transcription promotes lysogeny. When following the lytic pathway, lambda DNA directs the cell to synthesize components. Virions are assembled, the cell lyses, and the virions containing linear double-stranded DNA are released into the wild. The phage lambda attaches and injects its double-stranded DNA into the chromosome, made possible by a set of virally encoded proteins that allow the lambda phage to integrate into the chromosomal attachment site. The ends of the DNA join to form a circular double-stranded DNA, which then pairs with the cell's chromosomes. By contrast, in the lysogenic pathway, the double-stranded DNA recombines to integrate into the chromosome and become a prophage. The prophage is replicated, then distributed to progeny cells. Very rarely does the phage leave the chromosome by recombination to enter the lytic pathway. When it comes to the battle between the virus and the host, resistance to phage infection is possible. The host can battle against the phage using restriction endonucleases. These are bacterial enzymes that cut the double-stranded DNA carrying a particular palindromic nucleotide sequence. Being palindromic, the recognition sequence reads the same in the 5' to 3' direction of both DNA strands. The cut sites are staggered since there is a specific cutting site based on the DNA sequence itself. This results in DNA fragments whose ends have base pair complements. These are known as sticky ends and could be released by an enzyme called DNA ligase. The restriction enzyme system protects E. coli from infection by cutting the DNA that carries the sequence without cutting its own sequence, if it exists. To block the self-cutting of the DNA, there are often modification enzymes that will recognize the sequence on the host genome and chemically modify some of the bases. For example, a modification enzyme could methylate a base to chemically modify it. To reiterate, modification enzymes chemically modify bases to prevent the host genome site from being cut. This system acts like an immune system of sorts for bacteria to keep the virus at bay. Unlike our immune systems, though, the restriction enzyme system has no memory of past infections. It merely recognizes nucleotide sequences. The enzymes that cut DNA in this way have become important tools in molecular biology and gene cloning, such as the CRISPR technique. Also note that the restriction enzyme process can generate complementary DNA fragments that can be joined together by using specific DNA scissors, such as EcoR1. A plasmid DNA can be cut with the enzyme and joined to a fragment of DNA from any source. The ends are joined and the new recombinant DNA is inserted into E. coli to replicate it. The cloned fragment is now a part of the replicating plasmid inside the bacteria. Theoretically, you could make lots of this DNA by growing the bacteria in culture and separating out the plasmid DNA. This is how human genes could be cloned. Libraries of fragments derived from interesting sources such as human genomes could be built by storing every fragment in a plasmid of E. coli. The hard part would then be to find the fragment you want. CRISPR is a tool for gene editing. It is based on the natural system bacteria use to defend themselves against viruses. In CRISPR, the DNA cutting system is not limited to a single recognition site, and the restriction enzyme can have a memory of past infections. When foreign DNA enters a cell with CRISPR, the DNA is cut to inactivate the virus, but a fragment of the cut DNA is then inserted into a special location in the cell's genome. Fragments of the virus, or spacers, are arrayed like books in a library, with short repeated sequences in between to keep them separated. CRISPR makes it possible for a whole locus to be transcribed like a big bacterial messenger RNA. In this case, the big RNA is processed, so there are small RNAs that are responsible for targeting the CRISPR protein, called Cas9, to the sequence where it cuts the DNA, like the restriction endonuclease EcoR1. It is possible for this system to cut many different sites under the guidance of the stored CRISPR spacers. Complex of Cas9 and guide RNA can then cut any virus that tries to reinfect the host. Versions of the guide RNA can be made that will recognize any sequence that you wish, albeit with some minor restrictions. Thus, CRISPR provides a powerful tool with which to manipulate genes. For example, a version could be made to cut a gene that causes hereditary diseases in humans, then repair the gene with a normal copy. Another version could make specific DNA changes, such as changing one base to a different base, at a targeted site in order to modify genes in new ways or cure a genetic defect. This method could even correct inborn errors of metabolism. CRISPR raises the possibility of producing designer babies with traits chosen for their appeal, a true ethical issue that has raised many concerns about the development and use of the technique. We have spent a great deal of time discussing the viruses for bacteria, but what about eukaryotic cells? As you can probably already guess, eukaryotic cells are susceptible to a wide variety of viruses, with all the Baltimore classifications possible. Unlike prokaryotic viruses, many of the eukaryotic viruses are enveloped. Prokaryotic viruses skip the envelope feature because it makes little sense when attempting to enter a prokaryotic cell with a peptidoglycan wall. A eukaryotic cell lacking the peptidoglycan wall presents a membrane which the envelope virus can readily interact with and become fused with. Alternatively, the virus could be taken up by endocytosis after adsorption. To explain it differently, the three possibilities for a eukaryotic virus are 1. Have an envelope and fuse with the cell's plasma membrane to enter the cell. 2. Have an envelope and enter the cell by endocytosis. Or 3. Be considered naked by not having an envelope and enter the cell by endocytosis. Eukaryotic viral life cycles depend a lot on the genome of the virus. RNA viruses can perform their entire life cycle in the cytoplasm of the cell, whereas DNA viruses have the need to enter the nucleus of the cell so it can affect the DNA replication process. As already mentioned, eukaryotic viruses come in all the types that fall under the Baltimore classification. One such type are double-stranded DNA viruses. One example is HSV. The life cycle for HSV proceeds with the circularization of the genome and immediate transcription of early genes. Alpha proteins, which are the product of the immediate early genes, stimulate the transcription of early genes. The beta proteins, which are the products of the early genes, function in DNA replication to yield concatenameric DNA, followed by the transcription of late genes. The products of those late genes are gamma proteins that then participate in virion assembly. To summarize, gene products are made early and others made late during the life cycle of HSV, as well as some prokaryotic viruses. It makes logical sense to make some products early, since the enzymes for DNA replication or proteins can only enhance the synthesis of viral products. The later made proteins are likely to be structural proteins, since they aid in the virion assembly primarily. Once assembled, the virus seals its envelope from the nuclear membrane and is then transported out through the Golgi apparatus. A virus I'm quite confident that you have come into contact with before is the flu virus. The flu virus has the same life cycle as an enveloped single-stranded RNA virus. The endonuclease activity of the PB1 protein cleaves the cap and about 10 nucleotides from the 5' end of the host messenger RNA in a process referred to as cap snatching. The fragment is then used to prime viral messenger RNA synthesis by the RNA-dependent RNA polymerase activity of the PB1 protein. The viral messenger RNA is then translated, creating early products such as more NP and PB1 proteins. RNA polymerase activity of the PB1 protein synthesizes positive single-stranded RNA from the genomic negative single-stranded RNA molecules. The RNA polymerase activity of the PB1 protein synthesizes new copies of the genome using positive single-stranded RNA made in the third step as templates. Some of these new genome segments serve as templates for the synthesis of more viral messenger RNA, but later in the infection, they will actually become progeny genomes. Viral messenger RNA molecules transcribed from other genome segments encode structural proteins such as hemagglutinin, HA, and neuraminidase, NA. These messages are translated by endoplasmic reticulum-associated ribosomes and delivered to the cell membrane. Viral genome segments are packaged as progeny virions that then bud from the host cell. Interestingly, there are 3 distinct types of the flu virus, types A, B, and C. Type A is common in animals, with some versions capable of infecting human cells. For example, the swine flu is a combination of antigens for humans and pigs, making it quite a severe flu. The influenza from 1918 came about through antigenic shift, as well as the severe acute respiratory syndrome coronavirus of 2002, more commonly known as SARS-CoV. Each virus type has a distinct hemagglutinin, or capital H, and neuraminidase, capital N. These are proteins that are important for viral adsorption as well as virus release. Viruses have high variability in terms of the proteins that our immune systems recognize. This is why there is a new version of the flu every year and why doctors recommend getting the flu vaccine every year. Scientists making the vaccine have to guess which antigen on the virus will be the most likely that year and give those in the vaccine. Sometimes their guesses are wrong, which is why receiving the flu vaccine in a particular year may not actually protect you from that year's version of the flu. Though imperfect, it is the best chance of medical prevention available at the moment. Viruses can evolve in either small steps or big ones. Small changes that occur every year and change the surface antigens are known as antigenic drift. These changes are handled by changing the vaccine. However, if big changes occur, big problems arise. These big problems are created when two different viruses merge to create a completely new type of virus. This is termed antigenic shift. The new combinations of antigens have never existed together before, so an entirely new vaccine would be necessary, meaning loads more research on the new virus is required. Making a vaccine against a specific virus can be done in a variety of ways. The main challenge is to produce the antigens that your immune system will recognize and use to produce antibodies against the virus. The standard procedure followed in vaccine production is to first culture the virus in its appropriate host, purify it, then activate it. You would then inactivate it by either killing the virus and preserving the proteins associated with the capsid, or use a live virus that is unable to produce disease symptoms known as an attenuated virus. The live virus option is generally a better source for antibody production. In 1955, the Salk vaccine was developed to treat polio. It was initially a dead virus vaccine administered through injections, but was then eventually replaced by a live attenuated virus administered orally. The live version was developed by Sabin and replicated the gut to provide a better immune response to the infection. However, there is still a small chance of developing a polio-related syndrome from the Sabin vaccine since the virus is still alive, whereas the Salk vaccine has no such association. There are modern ways to make viral antigens that involve the use of recombinant DNA technology to make viral antigens in caterpillar cells. These antigens have proved to be better for vaccine production than those produced using chicken eggs. Let's talk a bit more about viral infections now. There are 4 possible results if infected by a virus. The first is developing an acute infection. In an acute infection, the virus causes cell death and is released over a short period of time. A persistent infection is the second possibility. Lasting longer than an acute infection, the persistent infection does not lead to immediate host death. Persistent infections include both chronic and latent variations. Chronic infections release the virus from infected cells for long periods of time, while latent infections remain within the cell for some time, only releasing the virus a while later. The fourth result of viral infection is a phenotypic change. A virus-dependent alteration of the host cell can lead to this phenotypic change. This could be the transformation of a normal cell into a cancer cell, for example. Viruses are not released from these cells. Instead, the viral genome acts as a mutagen by inserting itself into the host genome. If the right genes become activated or inactivated by this insertion, then the host's growth regulation can become perturbed. Like other viruses, plant viruses can be DNA or RNA based under the Baltimore classifications. Plant viruses are a bit different since the cells of plants are in fixed positions and separated by walls. Plant virus infections will only persist if the virus can cross into the neighboring cells in the plant tissue. In most cases, there are virally encoded movement proteins that help the virus move through microscopic channels between the plant cells, called plasmodesmata. The plasmodesmata is what typically allows plant cells to communicate with each other. An example of a plant virus that you may have heard of before is the turnip yellow mosaic virus. This next section of the episode will focus on human diseases caused by viruses. Something to keep in mind as we move into this part of the virology discussion is that prions are proteins that are infectious agents, but they are not viruses themselves. All right, on to the first virus, the coronavirus. The coronavirus is an enveloped, positive, single-stranded RNA virus. Its genome is enormous, featuring anywhere from 25 to 30,000 bases and 14 open reading frames. With such a large genome, it is capable of encoding for about 27 different proteins. 4 proteins that are important to know about are the spike, envelope, membrane, and nucleocapsid proteins. The spike glycoprotein is responsible for interacting with the host cell and is probably the one that will become the basis for modern therapies. As we know with the mRNA vaccines, they did actually go with the spike glycoprotein to produce that vaccine. The crown-like arrangement of those spikes is what gives the coronavirus its name. The structure itself is unusual, with polypeptides actually wrapped around each other. The receptor for this virus is a surface protein for ACE2, an enzyme attached to the outer surface of cells in the lungs, arteries, heart, kidney, and intestines. Infection succeeds in lowering blood pressure by catalyzing the cleavage of angiotensin II into angiotensin 1-3, with an activation step in there, of course. During infection, the spike protein is cleaved into subunits S1 and S2. S1 contains a receptor binding domain, RBD, which allows the coronavirus to directly bind to the peptidase domain of ACE2. S2 plays a role in membrane fusion. The West Nile virus is a group 4 positive single-stranded RNA virus. It is part of the flavivirus group because it causes encephalitis, an infection of the central nervous system. West Nile is primarily a bird flu, but it is carried by mosquitoes. It has an envelope but a small genome, and its life cycle is similar to that of SARS discussed earlier. The West Nile virus can infect skin cells and primary neurons. The Zika virus is a relative of West Nile, also mosquito-borne, and causes mild flu symptoms. Mothers that are infected with this virus can give it to their babies, leading to serious birth defects of the brain. The norovirus is another positive single-stranded RNA virus. Its formal name is the Norwalk gastroenteritis virus, as it spreads through the fecal to oral route. The major symptom from this virus is diarrhea. Norovirus is affected by temperature and makes the infected contagious from the moment one starts feeling sick to the first few days after he or she recovers. Unlike the other viruses mentioned, the norovirus is naked, meaning it has no envelope, and its genome is small. HIV is another virus many people have likely heard of before. HIV stands for Human Immunodeficiency Virus, but it is also known as HTLV, or Human T-cell Lymphotropic Virus. HIV is passed from mother to child and partner to partner. The origin of this virus has yet to be proven, but current hypotheses place its origin in Central Africa, where human-ape interaction is most likely. Immigrants from Africa to Haiti caused its widespread transmission there, then it subsequently migrated to North America. HIV eventually turns into AIDS, which can prove to be fatal for those infected. This virus is transmitted through blood, breast milk, semen, vaginal secretion, hemophilia, blood transmission, or perinatal exposure. Despite common misconceptions, HIV and AIDS are not transmitted through kissing, mosquito bites, the shaking of hands, or toilet seats. HIV is a single-stranded RNA retrovirus containing 2 RNA molecules in a capsid. The capsid attaches to the virus-encoded enzyme reverse transcriptase, where it then converts the RNA into DNA after entering the host cell's cytoplasm. The reverse transcribed DNA is duplicated to produce double-stranded DNA. Then it enters the nucleus and inserts into the host genome. It remains in the genome for many years while being able to express components of new virus. HIV has an envelope with inserted viral proteins, and it targets specific blood cells that carry a particular receptor protein called CD4+. Such cells are usually white blood cells that play important roles in the regulation of the human immune response. Thus the name Human Immunodeficiency Virus. It features 9,000 bases and has 3 major regions of expression: the GAG, POL, and envelope. So that's GAG, POL, and ENV. The GAG region encodes for capsid proteins, while the POL region encodes for reverse transcriptase, DNA integrations called integrase, and protein-cutting enzymes called proteases. The POL region is an important target for therapy. The envelope region is easier to remember since it encodes for envelope proteins involved in host cell interactions. These proteins are translated as a large complex that must be cut apart by the viral protease. The ends of the genome for HIV have long terminal repeats that play a role in virus integration. These are like repeated sequence motifs. HIV's life cycle starts with recognition of the CD4 plus receptor and subsequent binding. The virus then enters the host by membrane fusion, since the genome is uncoated. Reverse transcriptase converts the virus's single-stranded RNA into double-stranded DNA, which then enters the host cell's nucleus. The foreign DNA integrates into the genome and gets transcribed by the host's RNA polymerase. The RNA must then exit the nucleus to be translated into protein. As the viral RNA is processed, sections are spliced together and it is exported to the cytoplasm. The translated proteins are then processed by protease to become functional, and the virus is assembled. All in all, it takes about 24 hours to get from infection to virus assembly. Viral proteins are inserted into the host membrane, but the virus only acquires its membrane envelope when it buds off from the cell. This viral cycle does not cause lysis of the host cell, but it is capable of releasing 1,000 to 3,000 virions per day. The target cells eventually die by a form of apoptosis, and any lysed cell components released will cause more CD4 plus cells to congregate at the infection site. Proteins that interact with and co-recept with the CD4 receptor protein include GP120, CCR5, and CXCR4. Interestingly, individuals with mutant versions of the CCR5 co-receptor are resistant to HIV. HIV reproduces its genome by an error-prone process. Lots of variants are produced, and while some of them have high affinities for the receptor targets, many of them are non-functional. The high mutation rate of HIV makes vaccine production very difficult, and the integrated genome setup makes HIV nearly impossible to cure. Some researchers have proposed the use of CRISPR to remove integrated copies, but this has yet to be realized. HIV's course of infection proceeds as follows. There is the initial infection, which causes mild flu-like symptoms for the newly infected individual. Over a period of 12 weeks, the infection produces an initial burst of virus synthesis. Then there is a lower level of viral production happening consistently over a 5 to 6 year period, leading to a gradual loss of CD4 plus cells. At this stage, though, there are still sufficient numbers of CD4 plus containing cells to mount an immune response to the virus. Once the target cell level drops below the requirement needed to resist infection, though, the patient reaches an AIDS crisis. Normal, uninfected humans will have about 1,000 CD4 plus lymphocytes per microliter of blood, whereas individuals affected by AIDS will have less than 200 CD4 plus lymphocytes per microliter of blood. During the AIDS stage of infection, the infected individual is more susceptible to bacterial, viral, and protozoan infections, as well as being more susceptible to developing cancers. The virus may also cross the blood-brain barrier and wreak neurological damage, such as causing dementia. 4 types of pathological changes may ensue, including AIDS-related complex, abbreviated ARC, AIDS, AIDS dementia, or AIDS-related cancer. The AIDS-related complex exhibits in the form of mild fevers, weight loss, lymph node enlargement, and the presence of antibodies to HIV. The AIDS change leaves the person open to opportunistic infections, since they no longer have enough antibodies to prevent those infections. AIDS dementia is neurological damage as a result of the virus, but there is also evidence of other central nervous system damage possible. An AIDS-related cancer is Kaposi's sarcoma. This is caused by the human herpes virus 8, or HVV-8. This is a carcinoma of the mouth and rectum, with B-cell lymphomas. Ultimately, HIV causes suppression of the individual's immune response. Humans have a 2-part immune system. We have cellular-based immunity and humoral-based immunity. On the cellular side, white blood cells are activated by the presence of foreign antigens or cancer cells. Cytotoxic T lymphocytes, or CTLs for short, cause the destruction of cancer cells or virally infected cells to give the individual cellular immunity. On the humoral side, antibodies are produced by special white blood cells, called B cells, to protect against foreign antigens. CTLs and B cells must both be provided with information related to antigen presentation in order to be activated. In other words, they must be given a processed form of the foreign antigen to be able to understand their target. They also have to be activated by special cell chemicals known as cytokines that bind to the receptors before they can go to war against the foreign antigens. Both antigen delivery and cytokine activation are controlled by helper T cells, which are specialized white blood cells that help in mounting both cellular and antibody-based immunity. Unfortunately, AIDS kills the human immune system because the very T cells and other dendritic cells that are needed to launch the immune response carry CD4+ receptors that are the targets of HIV. As they are depleted, both sides of the immune system are lost. While vaccine prevention is tricky, chemotherapy treatment is possible. Though antiviral, chemotherapy uses drugs to target the viral life cycle at critical points. Such drugs are meant to target the virus-specific enzymes the human cells do not ordinarily contain, such as reverse transcriptase and viral protease. Without reverse transcriptase, the viral RNA cannot be transcribed to DNA. And without the viral protease, the proteins needed for virus production cannot be processed. Reverse transcriptase inhibitors are quite interesting. Reverse transcriptase makes DNA from an RNA template, as we already know. There are two types of reverse transcriptase inhibitors possible to prevent this, though. There's the nucleoside reverse transcriptase inhibitor, NRTI, and the non-nucleoside reverse transcriptase inhibitor, NNRTI. Though NRTI and NNRTI can work separately, they are more effective when working together. Essentially, they inhibit reverse transcriptase using anti-metabolites that look like DNA nucleotides, except that they cannot form polymers. Chemotherapy essentially targets the viral cycle that can be blocked. Other targets may also work, but they are expensive to research and produce, and so are not yet available. Examples of potential therapies include drugs that block viral interaction with cell receptors, drugs that block membrane fusion, and drugs that block virus genome integration. The HIV fusion inhibitor Fusion would be an excellent addition to current therapies if it wasn't so pricey. Through chemotherapy treatment, affected individuals can maintain a level of CD4+ cells sufficiently high enough to continue mounting an immune response. Chemotherapy merely controls the disease, but it is not a cure. The person will still be HIV positive and must take precautions so as to not transmit the virus to others. Not to mention, chemotherapy is expensive, so infection by HIV places a large financial burden on infected individuals. We have spent a considerable amount of time on HIV, so let's move on to some other viruses. Next in the lineup are tumor viruses. Unlike bacteriophages, tumor viruses alter the growth regulation of the cells they infect, meaning that instead of producing new viruses, they cause cells to transform into more tumor cells. This is achieved by integrating into the host genome and altering the expression of the host genes. In normal, healthy cells, signal pathways regulate the number of cells that can be produced. But for tumor cells, if the product of the genes they alter are regulators of cell growth, the pathway that inhibits excessive cell replication can be profoundly affected. Or, on a similar vein, the levels of growth promoter gene products could be increased, leading to the same result of increasingly more tumor cells being produced. Viruses associated with cancer are usually double-stranded DNA type viruses, but retroviruses do also exist, and they go through a DNA intermediate that can integrate into the genome. A few tumor viruses you may have heard of before are Epstein-Barr, hepatitis B and C, the human herpes virus type 1, HPV, and human T-cell leukemia type 1. Of those infamous diseases, Epstein-Barr, hepatitis B, herpes, and HPV are all DNA viruses, while leukemia and hepatitis C are RNA viruses. There is currently a vaccine for HPV. It is typically administered before the possibility of an HPV infection occurring. Because HPV is a cervical cancer passed through sexual contact, the HPV vaccine is often given around the age of 11 or 12, before adolescents begin to engage in sexual activity. This recombinant HPV vaccine administered is called Gardasil, if you were curious. Another virus you may or may not have heard of is the chicken sarcoma virus, or RSV. Francis Peyton Rouse is credited with the discovery of this virus in chickens with tumors in their legs. He found that by injecting those tumors into healthy chickens, new tumors were produced. Even the filtered material still caused an eruption of tumors. The transformation of chicken cells happens rapidly when infected by RSV. RSV is a retrovirus that has the gag, pole, and envelope genes we discussed earlier, but it also has an SRV gene that is not normally carried by other retroviruses. The presence of the SRC oncogene in RSV makes it unusual. Through trial and error of deleting certain genes, researchers found that if the SRC gene was deleted in RSV mutants, no tumors would form. Thus, the SRC gene is required for tumor formation in chickens. It is the cancer gene. Note that the SRC gene is conserved among all eukaryotes, making it a fundamentally important gene. It is fascinating that the viral SRC gene found in RSV is nearly identical to a normal gene in the chicken genome. This means that the virus must have become integrated with its host, then picked up the chicken gene before new viral genomes were made. Normal SRC genes do not cause tumors like the viral SRC gene does. Further studies revealed the viral version is a mutated gene. The enzyme tyrosine kinase produces this mutated viral version by putting phosphates on tyrosines in proteins. Unlike normal cell SRC, viral SRC does not have a built-in self-regulatory carbon terminus that can be phosphorylated to shut off the enzyme activity when the protein is folded a certain way. Different kinase regulators determine whether SRC is on or off. Since the kinase produced by viral SRC lacks the regulatory carbon terminus, the kinase cannot be shut down and is always active, meaning the cell will continue to divide. This whole process shows how signals that are passed through cells are very important to the fate of the cell. The signal is passed from the cell's surface to its nucleus by a series of kinases that activate more kinases. Normally, each signal must be active for a short time to reproduce cells, then turned off to regulate runaway growth. But in this case, cells continue to divide because the signal to reproduce cells is always active. Through this whole discussion, I hope you can understand why cancer is a genetic disease that includes both tumor-promoting and tumor-suppressor components. RSV is an extreme example because it has the SRC oncogene and tumor growth is rapid. Most tumor viruses do not carry oncogenes, and the tumor virus-induced transformations in cells require a much longer time to be realized, often years post-infection. Many genetic changes in multiple genes are usually required in a cell before it starts behaving as a tumor. The additional mutations required for tumor progression happen randomly over the course of a person's life. Though exposure to mutagens and the inheritance of mutant genes, or oncogenes, can reduce the time it takes to build a sufficient army of transformed cells. In general, though, cancer is considered to be the disease of older people because they have had the time to acquire the mutations. Interestingly, the human genome has a retroviral graveyard. About 8% of the human genome is filled with retrovirus DNA fragments. These are past viral infections of gamete-producing cells that cause retroviral genes to be passed from parent to child. Keep in mind, though, that these are incomplete viruses, mere fragments with a viral origin. Cancer-causing viruses are diverse in the mechanisms employed. As we saw with RSV, viruses can carry oncogenes, the cancer-causing genes. RSV just had one oncogene, but viruses could carry multiple. They could also insert a promoter or enhancer next to a cellular oncogene to cause an abnormal expression of that gene. For example, the virus could insert an enhancer that deregulates cell growth, or viruses could produce a regulatory protein that activates the cellular oncogene. The Epstein-Barr virus mentioned earlier is a prolific virus. A type of herpes virus, Epstein-Barr has been linked to mononucleosis, nasopharyngeal carcinomas, and Burkitt's lymphoma. This virus acts geographically. For example, people in North America are likely familiar with mono, while people in Africa would be more familiar with Burkitt's lymphoma. For those that don't know, Burkitt's lymphoma is common in regions of Africa with high incidences of malaria. The plasmodium that causes malaria suppresses the host's immune system, leaving those infected more susceptible to the disease. It is a cancer of white blood cells that causes swelling in the lymphatic tissue in the neck. It is common in childhood and can be quite aggressive. Another cancer-causing virus that relies on immunosuppression to promote tumor formation is HIV. Inflammation is another method viruses could utilize to produce tumors. Diseases such as hepatitis B and C, HPV, and herpes are suspected to contribute to tumor formation by causing inflammation. During inflammatory conditions, cell growth is often stimulated to help the organism fight the infection by providing better numbers of fighting cells. Viruses could take advantage of the increased cell production rate and act synergistically with any genetic changes that take place. A real hijacking of the immune response. Several neurological diseases plague the mammalian world. One such disease is the Creutzfeldt-Jakob disease, or CJD for short. This is a rare brain disorder that usually leads to death. It is caused by a mutation in the PRNP gene that is located on the short arm of human chromosome 20. The PRNP gene encodes for prion proteins, which are highly conserved proteins among mammals. Prion proteins are expressed most prominently in the nervous system. CJD is an inherited disease, and like I already mentioned, is exceedingly rare. The brain takes on a sponge-like appearance, and neurodegeneration leads to loss of muscle control, dementia, and death ultimately. Another neurodegenerative disease involving prions is Kuru. Kuru has a sponge brain phenotype and is specific to the tribal people of New Guinea. It has been linked to the funeral ceremony ritual, in which women and children eat the dead body, including the brains. Children are often the most affected by extensive neurological degeneration as a result. Scrapie is a neurological disease that affects goats. This spongiform disease leads to the loss of body function and eventual death of the goat. There is also bovine spongiform encephalopathy, or mad cow disease, more familiarly. This is a fatal spongiform disease that causes cattle to lose their bodily function. The disease is slow to express itself, taking between 2 to 8 years before symptoms are visible. As we saw with Kuru, consumption of meat carrying the disease puts others at risk. In the case of humans eating beef that's been affected by the mad cow disease, they can get a form of spongiform encephalopathy that is remarkably similar to the Creutzfeldt-Jakob disease, termed new variant CJD. Thus, humans can either get CJD through inheritance, as we saw earlier, or they can get new variant CJD through non-genetic means. The root causative agents of the neurodegenerative diseases I mentioned are prions. Please take care to note that prions are infectious proteins, not viruses. Prions are known scientifically as PRP proteins, of which there are 2 forms. There is the normal version, written PRPC, and those leading to neurological conditions termed PRPSC. The mutant form is misfolded, but still an extremely stable protein that is resistant to heat. When both the normal and mutant forms interact, the normal is converted to the abnormal form. As we already learned, prions can be ingested by consuming meat. Abnormal forms of the protein survive the digestive system and have the potential to cause neurological damage if they come into contact with normal PRP proteins. Ultimately, it is the assemblage of misfolded protein tangles in the brain that is an aspect of neurodegenerative diseases. It is an undisputed fact that humans have had contact with other animals. As the human population increases, cities expand, more land must be cleared, and contact with animals is made. Viruses from those non-human mammals then have the opportunity to infect humans. Variations of their variant genomes can lead to viruses with new capabilities our immune system has not encountered before. So, if you've ever wondered why there seems to be a new disease every few years, that's why. Keep in mind, though, that not all causative agents are viral in nature. Bacteria is still a prominent culprit in emerging infectious diseases. With increasing populations comes the increased industrialization I already talked about. Poor sanitization practices allows bacteria to culture in higher numbers than usual, leading to increased opportunity for bacterial infections. We have spent nearly the entire duration of this episode talking about viruses themselves, and not nearly enough about the work being done to prevent their spread or infections. So let's dive into vaccine theory to wrap up the episode. The theory of vaccines is to prevent the body with a germ antigen that cannot produce the disease with the thought that the body will develop antibodies against the antigen. Then, if the body does encounter the antigen again, the pre-developed antibodies will fight off the disease before it has a chance to make you sick. For this method to work, there needs to be a way to get foreign antigens into the body without causing harm, in order to elicit an immune response that will serve to protect against future infection. There have been a few different types of vaccines produced in accordance with this theory, the first of which that we will go over are live vaccines. Live vaccines are attenuated, meaning a weaker version of the germ or virus is used. Keep in mind that sometimes these inactive variants will appear on their own. Two rules that must be followed are that the live vaccine must reproduce to produce an immune response, but must not be capable of causing the illness itself. Examples of live vaccines include those for measles, mumps, rubella, varicella, polio, and chickenpox. Live vaccines are highly effective because out of all the vaccine types, they will have the best antigenic features and will replicate in host cells. Killed vaccines are different in that only pieces of the germ wall, such as sugars or proteins, are used in the vaccine. Formaldehyde is generally used to kill the rest. These types of vaccines are generally not as effective because they cannot reproduce. Hepatitis A, polio, rabies, and flu vaccines are all examples of killed vaccines. Another type of vaccine is the conjugated vaccine. These vaccines use the germ sugar attached to a protein to enhance the body's immune response. While it allows pieces of a virus to be used, it could potentiate the virus by adding immune system stimulators. For this reason, these types of vaccines are less common. Recombinant vaccines are more focused on genes than the virus itself. A gene from the disease organism is used to produce an antigen in the cell source it is designed to. Once the antigen is made, the rest of the virus can be dispensed of. That antigen is then injected in the vaccine. With this method, it is possible to make both viral and bacterial vaccines. Shingles, HPV, pneumococcal disease, hepatitis B, and Hib vaccines are all recombinant vaccines. The newest vaccine technology skips the bacteria or virus itself and instead focuses on providing instructions to the cells. The vaccine type I'm referring to is the mRNA vaccine. As the name suggests, these vaccines use synthetic messenger RNA to provide instructions for the cell to produce proteins the immune system will respond to. With the immune system responding to this benign threat, it will develop the appropriate antibodies to fight against the infection if it does ever enter the body. Note that mRNA vaccines are not meant to replicate the virus or alter the genome. mRNA by itself is not stable enough to be injected, nor could it enter cells on its own. To remedy this, a nanoparticle composed of lipids, essentially a fat vessel, is employed as its vessel through which it can enter cells. Thanks for listening to this episode of Fall Asleep to Science. Be sure to check out other episodes to keep the learning going.